Okay, so after that video, which if you're watching this recording, if you go on Canvas, there are two links to two videos. One is called Growing Body Parts, and that's what we just watched here in class. And this is a follow-up lesson. All right, so this is going to be called Stem Cells and Cloning. So let's go over some of the basics. And sexual reproduction, you have a sperm cell. Is that cell haploid or diploid, everybody? It is haploid. And for humans, that would be 23 chromosomes. Well, then we have the egg cell from the female. Is that diploid or haploid? Haploid. And for humans, that would be 23 chromosomes. And in sexual reproduction, you basically have one half of the dad, you have one half of the mom. It will result in a zygote. What is the zygote, diploid or haploid? Diploid. And the zygote would be one whole. And by the way, all these cells have a nucleus, so let's be sure to draw that. Well, after that zygote has been formed, a little while later, it is going to start to go through division, which we know of as mitosis. And eventually, it will become a little ball of stem cells called a blastula. Now, this is where you're going to get some new information. This blastula is made of non-differentiated cells called stem cells. Stem cells are non-differentiated, which every year, a lot of students don't know what this means. So I want to try to do a better job of explaining what it is. Stem cells are not different. Ben, is a brain cell from your brain different than a bone cell in your thigh?
Yes.
Yes. No, I didn't ask about the DNA. I just asked about the function of the cell. Yeah. A brain cell is different than a bone cell. Key word, guys, different. Mary, is a blood cell different than a lung cell? Mary, lung cell, blood cell. Yes. Now, I think you guys are hearing me say something different. This is why I need to make clarification. Is the DNA in a lung cell different than a blood cell? No. But is the function of a blood cell different than a lung cell? Yes. All right. So these stem cells are non-differentiating. We call that, and what they also are, pluripotent. Oops, spelled that wrong. Pluripotent. Pluripotent means they can become anything. They can become brain, they can become bone, they can become blood or lung or liver, kidney, you name it, stem cells can be it. Now, over time, as this little blastula grows, it will start to differentiate. Let's say that it's going to, and this is, again, this is called differentiation. Differentiation is a verb. It is a process. Let's say that some stem cells become neurons. That's found in your central nervous system. Let's say that some stem cells become muscle fibers. Muscle fibers are just really long cells of your muscular system. That's why they're called fibers. They're just really long. And let's say, for example, that some become a macrophage, which is a white blood cell. Are these cells different from each other? A white blood cell, a neuron, and muscle fibers? Yeah, they're all different. They have very special function. These cells are called specialized. Specialized cells are very good at a specific task. Do any of you watch or ever watch the show on E called Botched? where people have plastic surgery problems and these two doctors fix the problem. Well, one of the doctors is really good at reconstruction and plastic surgery of the body, basically everything from the neck down. The other doctor is really good at facial reconstruction and facial plastic surgery. So they both had their own specialties, and together they make a great little team. Well, the doctor that works on legs and torso and arms and fronts and backs, he's really not going to work on the nose or the lips or the cheeks. That's the other guy. Because the other guy, I think his name is Dr. Nassif, his specialty is the face. He specializes in the face. Do any of you in here play a sport? What do you play? Okay, there's only one position in tennis, so I'm going to ask someone else. What do you play? Basketball. Okay, what position do you play? Shooting guard. Shooting guard. How do you think you would do at center?
Probably, I'm short, so.
Okay, if you guys are unfamiliar with basketball, center is typically the really, really, really tall person on the team that hangs out under the basket and tries to get the rebounds, like Shaquille O'Neal, 7'2". LeBron James is 6'8". He's not even a center. He's not tall enough. Go figure. But what I'm getting at is Zoe's specialty is not being a center. Her specialty is being a guard. So everybody has their own little place. These cells are specialized. Stem cells are not. They are not specialized. They can become something specialized, but not yet. And so what scientists have learned is if you place these stem cells into an area of the body that needs them, like maybe because of an injury, you can instruct them to become whatever cell you want them to be. And this whole conversation of stem cell research really got started with an actor. And this actor was, and I'm going to share the screen with you guys at home. This actor was Christopher Reeve. Now, if you don't know who Christopher Reeve was, your age, you probably wouldn't know. He was the original Superman actor from the original Superman quadrilogy, Superman 1, 2, 3, 4. He was the man, Superman, America, in the late 70s and the 80s. Well, Christopher Reeve, R-E-E-V-E, he had an injury. He fell off his horse, and he became a quadriplegic. And so it was really disconcerting for people. Superman is indestructible, and he's paralyzed. Well, he was a huge advocate for stem cell research because stem cell research could potentially heal his injury. Typically with human biology, when you damage your spinal cord, it's not going to heal. It's permanent, which is why people who are in a wheelchair tend to stay in that wheelchair. However, with the invention of stem cell research and technology, perhaps those stem cells could have been implanted in his neck and heal his wound and basically give him a brand new spinal cord. But he died in 2004, and he never got to realize the advances that we have achieved with science and technology regarding stem cells. And so the big issue in those days was the source of stem cells. The best source of stem cells is embryos. They're called embryonic stem cells. The best source for stem cells are embryos. And in this country, abortions are legal. That was what Roe versus Wade was about. I think that was in the 60s or 70s. I'm not an American history teacher. But the best source of stem cells were embryos, and the only way you could really get embryos is through an abortion. And no matter what your stance is, whether you're for it or against it, that's your choice. People in this country were really up against it, and there were laws that prevented you from using. There were no laws against abortion, but there were laws against what you can use the aborted embryos or fetus for. Now, I want to just give you some facts. When you are a blastula, like this right here, you are a bundle of undifferentiated cells. There's no heart cells, there's no brain, there's no nothing. It's just DNA, really. And so whatever your opinion is on when life starts, that's your opinion. But just as far as the facts are, these cells are not specialized yet. But nonetheless, back in the 90s and the early 2000s, you could not use the embryos for research. You couldn't use it for therapy. And so there's a big argument about, the people that were in favor of embryonic stem cell research and therapy said, these people like Christopher Reeve, they're here right now, and they're injured, and they need treatment, they need help. And the people that were against it would say, Well, you're taking a potential life away for someone who hasn't existed yet. You're not giving them a chance. Christopher Reeve had his chance. Now let's give this potential person a shot. So it goes back and forth. But in the last 10 or 15 years, the laws in this country have changed to where we can actually use embryonic stem cells, but also we found other sources for stem cells. Other sources are mainly bone marrow. There have been some that can be found in the brain. I'm really not going to cite that. Those aren't really that good. Bone marrow stem cells are okay, but they're not the best. They can become other cells, but there's a limit to what type of cells they can be. Embryonic stem cells can become any cell in your body. Bone marrow stem cells have a limit, like maybe 10 or 12 different types of cells. Yes, Natalie?
Why is there stem cells in bone marrow?
Because that's where your blood originates from. So you have to be able to take stem cells that are nothing, and they have to become things like white blood cells, red blood cells. So throughout your whole life, you're still generating blood cells. They need to be able to come from somewhere. So you need to have the original cells, and they need to be instructed to grow into red blood cells or white blood cells or lymphocytes or leukocytes. Okay? So nowadays, I'm driving home from plant, I'll hear a commercial on the radio about Dr. Locke's stem cell research, best stem cell therapy. My name is Derek Brooks. I'm a former linebacker for the Tampa Bay Buccaneers, and I had Dr. Locke's work on my wrist, and he gave me stem cell therapy. Now my wrists are as good as new. I advise you to go see Dr. Locke's for all your stem cell needs. I mean, it's advertised on the radio, and it works. You are basically given like a newborn, new cells. And so that is going to get me to the second part of today, which is cloning. The one problem that we have is your body rejecting tissue. But what if it's your own cells? And so here's what we're going to do. The steps for cloning is actually very simple. The implementation of it is more difficult. Do any of you know what animal was cloned in the 90s? It was the first animal cloned in history of science that we know of. Dolly the sheep. Very good. Well, Dolly the sheep took about 276 tries to get it right. You're like, what do you mean 276 tries? There were, let's say, defects. It didn't grow to term. It was very sick. It basically was born. It was like, kill me. It was totally messed up. Like 200 plus times. They couldn't figure it out. And then finally, on try number 200 and blah, 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 Dolly the sheep was born, and it was fine. And that was a huge breakthrough. Well, in order to make a clone, first off, you need to obtain an egg cell from the same species that you're going to clone. So let's say I'm going to clone Ben. I need an egg cell from a human. Ben's a human. I need a human egg cell. So here's our egg cell from Miss Who Cares? It doesn't matter. As long as the egg cell is good. And you all said earlier that an egg cell is haploid. So I'm going to put the letter N in there, which represents haploid. Now, step 2, remove the nuclear DNA. So what you do is you stick a little needle, like a microscopic needle, into this egg cell, and you suck out the DNA. So, you ready? Here it goes. Out. So now this egg cell is blank. It's a blank. And then, step 3, obtain the full DNA of the subject to be cloned. So if I took a cell from Ben's wrist, that's a skin cell. Does his skin cells have all of his DNA? Yes, they do. And so I could just take one of Ben's skin cells, like right here, and inside that nucleus is all of his DNA, 2N. And here's how we do it. We then, step 4, remove the somatic cell DNA and implant it into the egg cell. So we poke this skin cell of Ben with the needle, we suck out his DNA, And then we put it into this egg cell. Now, normally, guys, in order to fertilize an egg cell, you got to have half of dad, half of mom, half and half makes one whole. This is totally different. We have now given an egg cell a diploid nucleus. This egg cell now has 100% of Ben's DNA, not 50. This is all of it. And then here's the weird part. Step 5, you shock it. You zap it. You zap it to stimulate mitosis, to get it to grow. And let's say, oh my gosh, it's growing. It's going through mitosis. Here we go. We have a blastula. Oh boy. And what is this blastula made of? Stem cells. And guess whose stem cells they are? Ben's. His DNA. And now, here comes the crazy part. These are Ben's stem cells. We can do one of two things with them. Let's say that we want to make a clone of Ben. This is called reproductive cloning. A lot of the countries in the world have deemed this to be unethical, but we've done it with plants, we've done it with animals. We use a surrogate mother. We will implant the cloned cells into a surrogate mother. And then 9 months later, if everything goes right, you have a clone. It's going to be a little baby Ben clone. But Ben, how old are you? So he's 17. This DNA that's in the clone is aged 17 years. So let's say hypothetically Ben is able to live to 90 years old. His clone will live to 73. The clone is brand new. It's a baby. But the DNA is 17 years old. And so it's like, that's what we learned with Dolly the sheep. It did not live a full life. So that's one possibility. And then the other possibility is therapeutic cloning. Let's say Ben had kidney failure and he lost his kidney. It's gone. Well, we can instruct what you do is you keep the stem cells outside the body. You do not put them in the mother. You do not put them anywhere except in a lab. You keep the stem cells outside the body and allow them to replicate over and over and over. And so you're going to just get, tons and tons and tons and tons and tons and tons of cells. Lots and lots and lots and lots and lots and lots and lots. I can do this all day, running low on time. Hope you get the idea. And what you can do is you can instruct the stem cells to become any type of specialized cell. So Ben had a kidney disease. We will instruct these cells to be kidney cells. And as you saw in the video, we will put these kidney cells into a mold of a kidney, and the cells will grow into a kidney. And then we bring Ben in for surgery, and we put his kidney inside of him, and his body won't reject it. So that is known as therapeutic cloning. All right. That is it for today.